Well, thank you for inviting me to participate in this really cool meeting. I appreciate it. This is going to cover a lot of stuff quickly. We're going to go through several different mechanistic aspects of how proteins misbehave and how they're stabilized under various stress conditions. This literally goes back more than 30 years with collaborations with people from all over the place. This is just a few of them. But here's the idea. Here's the governing idea for this talk, and hopefully for the way your career should be looked at. And Rena is the perfect example of this. You got to learn the basic and fundamentals to master the practical and applied. And if you don't, you're going to run into problems all the time. And we see this all the time. And all of us have had cases where we didn't really understand what we were doing as well as we should have and got some nasty surprises. So I'm going to apply that idea to protein aggregation and stabilization in solution during freezing and freeze thawing, during freeze drying, and at the end at interfacial stress. So before we get into the mechanisms, let's talk about practical. These are two real world case studies, and I want to go through them first and then have a question for you, a little quiz to start with, and we won't require you to answer it publicly, but you can jot down your answer. And then later, after we go through some relevant mechanisms, we'll come back to this. Here's the situation. Case one is an antibody bulk drug substance was thawed, and it was very cloudy. The protein had precipitated. That precipitate was collected by centrifugation immediately after thawing. And as Rena said, you can use IR to look at anything. So what's very cool is you can take a precipitated protein and get IR spectra of it. And in this case, the protein molecules in the precipitate had fully native structure. Also, if an uncentrifuged sample was left out at room temperature for a while, the precipitate dissolved and went back into solution. So this is a case of reversible freeze-thawing induced precipitation. The second case, everything looked the same. Upon thawing, there was this cloudy solution. This solution was held at room temperature for a while, and the precipitates just settled to the bottom. They never redissolved. IR spectra were obtained of the precipitate, and it showed extensive intermolecular beta sheet structure of the protein molecules because the precipitate had protein aggregates. So this is a case of irreversible precipitation. Okay. So first thing, at least this level of characterization, which isn't always done when people face precipitation, this level of characterization gave some insight into one case being reversible and the other's irreversible. So here's the quiz. In which case would inclusion of 0.3 molar sucrose reduce the amount of precipitation? Another way to ask this is adding sucrose a good formulation strategy for either of these cases? So you can jot down your answer. Do you think it's case one, case two, both cases, neither case, or the last one, which we all love, no way to know without hiring consultants and further root cause analysis, right? That's usually what, by the way, that's how come I have these cases to talk to you about. These are real world case studies. So is it case one, the reversible precipitation? Would that be helped by sucrose? Case two, irreversible, both or neither? And we'll come back to this after we go over some mechanisms. Now let's talk about some very important protein stabilization mechanisms. These are used all the time in your formulations, whether you know it or not. And the first one's very simple. It's a manifestation of what we call the Wyman linkage function. If you don't know what that is, look it up. And there's a whole book and hundreds of papers on this. But let's look at what people use in the industry practically to do formulation screening. This is an ancient study from David Volkin and colleagues. And they were simply using fluorescence spectroscopy, doing some cooking studies. So heating up this protein, which is fibroblast growth factor, without any stabilizer, very low transition temperature. You can see that here. And then when they added in this case, heparin at a certain mole ratio, they had a massive increase in stability as indicated by these thermal scans from fluorescence. And then if you plot these data, this is a remarkably unstable protein, and it's hugely stabilized by stoichiometric levels of heparin. So heparin here is a strong binding ligand. It binds strongly to the protein and provides a great deal of stability. Well, how does that work? Well, if you look at this simple free energy diagram, if you look at native protein and denatured, obviously native has a lower free energy than denatured. And if you add a ligand in, heparin here in the example, the protein ligand complex has a much lower free energy than the protein alone. This complex wouldn't form unless there was this free energy decrease. The heparin doesn't bind to the denatured state. So by throwing in heparin, you greatly lower the free energy of the native state. You don't affect the denatured state. Therefore, the barrier between the two states is much greater. And that's reflected here in this increase in the apparent thermal transition temperature as a function of heparin. So strong binding ligands can give you remarkable stability. And say you have a protein, a mAb, or one of these new Franken molecules, and you really don't know what might be a good ligand. You can find this out indirectly by doing high throughput cooking studies. There's a number of methods now that allow us to look at a lot of samples quickly and find out, do any of the compounds you added have this big effect on thermal stability? And you can't do this if you just screen a very limited group of excipients. You have to do a broad screening. And sometimes you get lucky. I know of a case of an antibody, calcium binding to it greatly stabilized it, kind of this effect. And unfortunately, that's usually not done. Most people are not doing broad enough screenings to get lucky to find strong binding stabilizing ligands. What they're doing instead is doing non-specific stabilization. And we understand that, thankfully, because of the remarkably great work from Professor Serge Timoshev, one of our wonderful friends and mentors in the field. And what he looked at were non-specific stabilization mechanisms by compounds like sucrose and a number of other stabilizing additives. We call them excipients. He called them co-solvents. In biology, they call them osmolytes. We also call them cryoprotectants. All of these compounds have the same mechanism. And it's quite a bit different than the strong binding stabilizing ligand mechanism. First off, it usually takes a fairly high concentration, say 0.3 molar, to see stability manifest itself. What Professor Timoshev and his postdocs and students found out is sucrose is actually excluded from the surface of the protein. That is, there's a deficiency of sugar around the protein compared to the bulk phase. If you think about that, not from a molecular standpoint, but from the thermodynamic standpoint, that's negative binding. So if positive binding between an additive and a protein reduces chemical potential, negative binding increases chemical potential. This is the big take-home message. Stabilizing additives, excipients like sucrose, increase chemical potential of the protein. Well, that's interesting. How does that increase stability? Well, the degree of exclusion depends on the surface area of the protein species. And obviously, an unfolded protein has a much higher surface area than the folded. So the increase in chemical potential is much greater for the unfolded protein than the folded. So if you think about it, if it's much greater increase in chemical potential compared to here, look at it from a thermodynamic box. So you have your normal in water situation. Here's a native and here's the denatured protein. You have a certain free energy barrier. We put the protein into sucrose, its chemical potential is increased some. But if we put denatured into sucrose, its chemical potential is increased a lot more. So because of the greater chemical potential increase for the denatured, the barrier between the two states greatly increases. Another way to think about this is from the principle of Le Chatelier, and can be applied generically to any situation. And in this case, the principle says if you perturb the thermodynamics of the system will tend to minimize that perturbation. What that means here is sucrose increases chemical potential, the system's going to want to minimize that. And so the equilibrium will be shifted toward the native protein. And this is really an important way to think about it. We're going to favor the most compact native species in the population in the presence of something like sucrose. And what that does from a folding standpoint, if we look at unfolding as a function of guanidine, your typical curves, in this example, as you get more and more sucrose, you're taking more and more guanidine to unfold the protein. You're increasing the thermodynamic stability. And that was even shown with temperature scans for this protein. But what about what we're most concerned with? We're not really worried so much about the equilibrium between unfolded and native protein. 99.999% of our protein molecules are native in our formulations. What we're worried about is aggregation. And that's shown here in a simple pathway. Your native proteins in equilibrium with species that are very slightly tweaked in structure, partially unfolded species, which have the capacity to aggregate. Sometimes they do, sometimes they don't. But the higher level of these species, the more rapid aggregation will be. If we put sucrose into a formulation, we shift the equilibrium away from partially unfolded toward fully folded molecules. And that's shown here in this reaction coordinate diagram for aggregation. You have a native species going to form a reactive species, a partially unfolded molecule, and there's an energy of activation for that. Then that falls over and finds a colleague and eventually makes aggregation. In the presence of sucrose, this activation energy barrier increases because preferentially excluded sucrose increases the chemical potential of a partially unfolded molecule more so than that of a fully native molecule. The other way to look at that is you're shifting the equilibrium away from these aggregation competent molecules toward fully native molecules that won't aggregate. This is what's happening in our formulations all the time when we have additives like sucrose. And here's an example of that, basically showing monomer loss of granulocyte colony stimulating factor very rapidly at pH 7. This protein does not like pH 7. And as you do the study as a function of higher sucrose concentration, you can slow down the aggregation. And you can get an insight in the shift of the molecular population qualitatively by looking at amine exchange studies done with IR. As you throw proteins from water to deuterium oxide, as the protein has deuterium exchanged for hydrogen, you shift this one band, and you can follow that shift as a function of time. And if you do it with no sucrose, it's relatively fast. And as you add more and more sucrose, the shift is slower. That's because the exchange occurs from partially unfolded molecules. And by putting in sucrose, the equilibrium in the molecular population shifted away from partially unfolded toward fully folded molecules. So we just talked about this. Aggregation is dependent on the transition of native molecules to partially unfolded. So structural or conformational factors are important. But that's not all that matters. And, you know, in the literature now, you read tons of papers about colloidal stability measured formally by measuring with light scattering and other methods, the osmotic second virial coefficient. Also, that kind of correlates with this parameter called KD, which people can measure conveniently with dynamic light scattering. But the point is, you can have an unfolded molecule, but for it to aggregate, it has to be colloidally unstable. And so pH is a key factor. If this molecule has a pH near its isoelectric point, it can have a very attractive interaction with the neighboring molecule and aggregate very easily. Whereas if you have a pH to where it's highly charged, charge repulsion will make partially unfolded molecules less able to aggregate. And so both conformational stability and colloidal stability are important. And here's a really good practical example. This is looking at the same protein, GCSF again. And in this study, under the conditions, here's loss of monomer due to aggregation during incubation, a moderate level of monomer loss. Sucrose kind of slows that down. If you put in benzyl alcohol, the protein just aggregates immediately. And that's because benzyl alcohol causes partial unfolding of a lot of the molecules. So the conformational stability is really tweaked and it just aggregates like crazy at pH 7. But at pH 3, first off, notice at pH 3, it doesn't aggregate in the absence of benzyl alcohol. The protein's highly charged at pH 3, and it's an unusual molecule at pH 3 in just buffer. It's actually fully native. You put in benzyl alcohol and its structure's perturbed at pH 3, just like it is at pH 7. So you have partially unfolded molecules that are highly populated, but they don't aggregate. The reason they don't aggregate is colloidal stability is dominating here. The highly positively charged molecules are incapable of aggregation, even though they're partially unfolded. So both conformational stabilizers like adding sucrose can help, but colloidal stability can be super important. So what do we do with this practically? Well, typically you don't have to measure conformational stability and separately colloidal stability. Most of the time, formulation screening for proteins is done by heating studies. You can have high throughput fluorescence heating, you can have high throughput light scattering heating, all kind of ways to do it. And you can look at a range of conditions like pH, buffer type, and excipients to find out what do they do to the apparent thermal transition temperature, so-called TM, or the onset of aggregation temperature. And what you find is as you have, say, a really good pH, that temperature will increase. And it increases both because of the impact on conformational stability as well as colloidal stability. All these heating methods are irreversible methods. That is, they cause the protein to partially unfold and aggregate during the course of heating. Therefore, both conformational and colloidal stability effects are manifested, which is great. I mean, that way you can actually just use thermal scans as a way to get an insight into the overall effect of an additive or a solution condition on your protein stability. So this is a super valuable method and obviously used all the time in the industry. So let's switch gears now. What happens during freezing? Are we going to have the same kind of problems and need to stabilize? Well, of course, proteins are unfolded during freezing. You get unfolding. You also have, in addition to low temperature, you have high salt and protein concentration. The pH may change. You have ice-liquid interfaces. So you have all kind of things that are perturbing the conformation. And also the colloidal stability is reduced because you're increasing salt concentration. And if you're doing freeze-drying formulations, damage occurring during freezing is going to carry over during drying. So how do we stabilize proteins during freezing? The same mechanism. When a solution freezes, the protein and the non-crystalline excipients are with the water. So sucrose, protein, and water are in the non-ice phase, and preferential exclusion still works. So Timashev's mechanism works for freezing also. And the amount of protection depends on the initial concentration, bulk concentration of the additive, just like it does in aqueous solution. So what about practical problems with freezing? Well, the number one problem I've seen over the years is people use sodium phosphate buffer in biological formulations that get frozen, say bulk drug substance for an antibody, AAV formulations used for gene therapy, you pick something. Sodium phosphate's a lousy buffer, and it's a lousy buffer because it tends to acidify during freezing. And that's because the basic salt will crystallize out, leaving maybe up to pure monobasic salt. That can be limited by kinetics. If you cool and warm quickly enough, it might not happen. It also can be attenuated by other additives like amorphous sucrose. But the practical advice is don't use sodium phosphate. But if you do, that's okay, because you're probably going to have to get help trying to figure out what's going on. And if you call me, I can get money for fishing trips, so it's all right. So don't use a bad buffer for freezing protection. If you optimize your solution conditions, say by thermal scanning, to pick the best formulation that works in solution, that will also help during freezing. And obviously, nonspecific stabilizers, so-called cryoprotectants, are useful. And importantly, non-ionic surfactants can be really good at reducing aggregation because a large fraction of the molecules are caused to unfold because of interactions of protein molecules with the ice-water interface. What about during drying? Well, proteins are what they are because of interactions with water. And we remove water, proteins unfold. Remarkably, in the old days, in probably the 1980s, there were fights about whether dried proteins were unfolded or not. There are a few professors on each side that had an excuse to get together and yell at each other and then go drink beer. Well, the one side that said they were unfolded was correct. And so what happens is now that you pull away water, the protein unfolds, you get non-native interactions, intra and intermolecular interactions. And these sort of interactions, of course, are fostered because the protein on its own and dry, there's nothing to separate protein molecules. And then when you rehydrate the sample, there's competition between refolding and aggregation. So proteins that are dried without stabilizers often aggregate like crazy upon rehydration. There's a few special ones. We call them the biophysicist friends, like chymotrypsin, an RNA that may not have a problem refolding upon reconstitution, but most of our drug proteins are not real happy dried without additives. And the other thing is your long-term storage stability can be greatly compromised if your dried formulation has unfolded protein molecules. So how do we protect during drying? Well, another group of us got in a fight in the 80s and 90s over mechanisms of protein stabilization during drying. One side said it's just glass-forming excipients, and they lock the protein in a lollipop and it can't unfold. And the other side said, well, yeah, it has to remain amorphous, the excipient does, but it's doing its job by replacing the water molecules that are removed from the dried protein. And there was a third group, and there still is, that claims that the sugar around the protein actually keeps a hydration shell around the protein. Well, at the level of drying we do commercially, there's no water left around the protein. A hydration shell of a protein molecule is thought to be around 0.3 grams per gram of protein, and we're drying to less than 0.01 gram per gram usually. So retention of water is not the mechanism. Glass formation is important, necessary, but not sufficient. The real mechanism is amorphous sugars hydrogen bond to the protein in the place of lost water. And this can be shown with IR spectroscopy. This is a protein with highly alpha helical structure, interferon gamma. The dotted line is for the native second derivative spectrum. The dried protein spectrum shown in a solid line is greatly perturbed because the protein's unfolded. When you rehydrate it, about 20% of it aggregates. So the rehydrated spectrum, you could even pick up a non-native intermolecular beta sheet band. If you dry the protein with sucrose, the solid line is for the protein in the dried state in the presence of sucrose. You get a much more native-like spectrum. It'll never be perfect. I mean, there is some intrinsic effects of water itself on the vibrational spectrum of a protein, but you can see it's much more native-like. You rehydrate it, it has a tiny amount of aggregate. By the way, IR is not a sensitive way to detect aggregation, but it's a really nice way to look at what's in the aggregate. You can spin these precipitates down and do structural studies and find out the precipitate, which in this case was only about 5%, is mostly intermolecular beta sheet. So sucrose, hydrogen bonds to the dried protein, replaces water. What about dextran? Well, dextran, this is 40 kilodalton dextran. It's a large polymer. It's incapable of hydrogen bonding to the dried protein due to steric hindrance. And what you see is when you dry a protein with just buffer, with dextran, these solid and other lines, there's no effect on stabilization. The dextran forms a wonderful glassy matrix, but that's not sufficient to prevent unfolding. So what about storage stability? As you look at numerous examples in the literature, what you usually find is as you increase the ratio of sucrose or trehalose to protein, you get better and better retention of the native structure at time zero in the dried formulation. And in this case, that's exactly what happened. This is interleukin 1 receptor antagonist work from Byung Chang and colleagues. All these formulations with different amounts of sucrose had about the same TG value, about 58, I think. And what you find is if you look at turbidity, a crude measure of aggregation, this protein's really not happy during freeze drying and storage without sugar. And as you add more and more sugar, at the resolution of turbidity, at least, you are inhibiting aggregation. And chemical degradation, in this case due to deamidation, is also much less if you have higher amounts of sugar. So having a native structure at time zero does correlate a lot with increased storage stability. And this has been seen in numerous examples. So as an overview, protein stabilization, it's either ligand binding or Timashev mechanism. Freeze thawing, it's the same thing. Drying, it's water replacement. And storage, you need a native protein and a glassy matrix with a high glass transition temperature. So let's return to our two case studies. And I'll just go over the answers and you can see if you had the same one. So case 1 was reversible precipitation due to freeze thawing. Case 2 was irreversible. Which case would sugar help? Well, it's case 2. The case 2 is a problem because the protein molecule is structurally perturbed during freezing, and upon thawing, it aggregates like crazy. So we need a structural stabilizer. A preferentially excluded excipient like sucrose would provide that stabilization. Case 1, interestingly, sucrose would make it worse. And, if we were in the room together, we could go around and ask why, but just think about why would sucrose make case 1 worse? Well, the reason for that is sucrose increases the chemical potential of the protein. That means the solubility of the protein is decreased. So if we throw in sucrose, the precipitation would be worse. Why? Well, reversible precipitation is occurring simply because the protein has its solubility limits exceeded during freeze concentration. And so its equilibrium solubility is exceeded, it simply falls out of solution. When you thaw it and the ice melts, you get it going back into solution. So it's already having a problem with equilibrium solubility. If we put in sucrose, we reduce equilibrium solubility even more, and it would make it worse. So with these two real-world examples, by having mechanistic insights into how additives work, you can pick the right strategy, okay? Here's a bonus question. What about if we had said, hey, in this earlier study, we found what we think is a really strong binding ligand that stoichiometrically binds to the native state and greatly increases stability. Would that help? Well, in case 2, where irreversible aggregation is occurring, surely it would help. You stabilize the native protein, you get less unfolding, you get less aggregation. But it would also help in case 1 because a strong binding ligand will reduce the chemical potential of your protein, which increases solubility. And in case 1, actually, from a practical standpoint, that would be a way to solve the problem. I don't think it was ever done that way, but it would work. Okay, so that's kind of your normal protein in bulk solution, freezing and freeze drying. But what we face often in industry is a whole other problem. And that is particle generation by interfacial stresses. These are some of the earliest MFI imaging, microflow imaging images published. This is for an antibody that was agitated. And if you look at these images, and a lot of us have looked at a lot of these images, these look like bits of film that were crumpled up, broken and crumpled up together. So think of an air bubble, or the soap bubble. Envision this as an air bubble in solution with protein molecules. The air bubble will be coated with a film of protein. And if that bubble pops, you generate these things. So what happens is proteins absorb to any kind of interfaces, and they form films or gels. Here's an example, actually a macroscope. Here's a picture of a film being stretched off the surface of an air-water interface. Here's an air bubble that we studied in my lab, and you can see protein absorbing and actually making clumps. And as you reduce the air bubble volume and then increase it, you mechanically stress that surface layer and it pops off. So what happens is you make films, and then anything that helps to disrupt and break those films leads to particles in solution. And obviously, let's say you have an air-water interface and you're shaking, the interface compresses and dilates, that knocks protein film into solution. Well, new molecules come in to replace it, and you have this continuous process. And so you get many different cases where layers are absorbed and make films and then break loose, like inside tubing used in processing for peristaltic pumps, layers absorbed onto syringes. You may have multiple interfaces, like an air bubble in a syringe, and even mechanical shock can dislodge these films. And here's a really cool study. This is from a few years ago. They just did a simple air-water interface stress. They had a protein solution in water, and every time they rotated the test tube, a needle would rupture the interface, or they had another case where the needle just sat in the interface and there was no rupture. And by light scattering, they showed that rupturing of the air-water interface breaks apart a film and leads to a lot of particles. And this all was inhibited with polysorbate. We've seen exactly the same kind of results using the same setup with silicon oil water interfaces. So definitely adsorption of films to interface is important, and the rupturing those films is really critical. So what about solution parameters, conformational and colloidal stability? Well, yeah, they may play a role. So here's an interesting study. This is from Danny Chow from years ago. And this is looking at a fusion protein, interferon fused with human serum albumin. And he was doing loss of soluble monomer by SEC as a function of time of agitation under different solution conditions. And at pH 7, this protein had an intermediate level of monomer loss, and at 5 and 6, it was worse. And at pH 3, even though you were shaking it and there's all this film formation and everything's potentially happening, it wasn't aggregating much. And if you looked at conformational or colloidal stability by B22 values, you find at pH 3, it's very colloidally stable. So even for some proteins, interfacial stress and the amount of aggregation occurring can be modulated by B22 or colloidal stability. And look here what happens with various excipients. Well, in buffer alone, this is loss of monomer at pH 7 again, a moderate colloidal stability. You throw in salt by charge shielding, you lose your colloidal stability and the protein aggregates more quickly. Here's a bizarre one. Sucrose accelerates agitation-induced aggregation for this protein. It does reduce colloidal stability, and there's some thoughts that sucrose does weird things to the protein at the air-water interface. And of course, the magic excipient is a non-ionic surfactant. It's not working by doing anything to conformational or colloidal stability typically. It's just competing with the protein for the air-water interface. And we'll come back to this mechanism in a couple of minutes. Here's something that we hear about a lot, those of us that work with various companies, is people will have particle problems. Usually it's visible particle problems. Normally subvisible particles aren't what leads to a panic call saying, oh my gosh, I've got a problem in my product. Visible particles like the ones here in this pre-filled syringe are a game stopper. They can happen during, even initial attempts at clinical manufacturing during fill finish. They can happen surprisingly in a well-established product that never had a problem before. And in many, many, many cases, the cause of these particles ultimately comes down to filling pump stress. And every kind of pump has various stresses. Rotary piston pumps and time pressure filling systems are really susceptible to cavitation. All of these systems have interfaces where proteins can form films that can be formed and rupture. You get it with all kinds of pumps. You can have particles shed, stainless steel from piston pumps and tubing particles with peristaltic pumps. And a really interesting one, which is another real-world observation, changing the pressure in a time pressure filler in the pressure vessel can lead to more outgassing and more bubbles or increasing the pressure can lead to more bubbles during filling and lead to particle formation. And this is a real world one that I worked with some friends on. And it was not hard to figure out, but it was kind of a mess because we had a brand new facility that had this higher pressure system. So, what can you learn about studying micro and nanoparticles during peristaltic or pumping studies? And this is an example of peristaltic pumping. These studies are all published now. I didn't put the reference, but you can find it. So this is from Cheng He, former postdoc. And what we did here is we're just looking at a model protein, intravenous immunoglobulin, formulated in a buffer where it's not real happy, PBS. So we want something to happen. And basically, over the time course of a 500 mil pumping with a peristaltic pump, you get different amounts of microparticles depending on the tubing brand. Now, that was kind of a surprise to us. We didn't realize this would happen. And even bizarrely, you can have different amounts of nanoparticles. So this one PharmEd tubing, pumping through it led to a lot of nanoparticles and less microparticles. And then you could do post-pumping agitation to see what those pre-existing particles might do to stability. And this one tubing that had a high level of nanoparticles actually generated way more microparticles during agitation. So those nanoparticles were essentially seeding microparticle formation and are agglomerating during agitation. And none of this would have been visible to most people because they don't look at nanoparticles, and they rarely even look at microparticles with this method, which was flow imaging. What about buffers? Well, for this protein, IVIG, in an earlier study, we already learned the commercial formulation provided a high degree of colloidal stability as measured by B22. And PBS, which we knew was not a good buffer for the protein, greatly reduced colloidal stability. So what does that do during filler pump operations? And what we found is, in general, glycine buffer formed fewer microparticles during pumping than the protein in glycine formed fewer particles than the protein in PBS. And with nanoparticles, interestingly, the PBS formulation for this one tubing made a lot more nanoparticles than the IVIG formulation. So unpredictable things can happen during pumping, and you just need to do the studies to find out what those things are. And then what about post-pumping agitation? Well, for microparticles in general, glycine buffer gave the protein way more stability in agitation after pumping. But again, with nanoparticles, here's this bizarre thing. IVIG and glycine buffer, agitation caused a big increase in nanoparticles. We're guessing that's because colloidal repulsion between nanoparticles kept them from aggregating as well during agitation than nanoparticles would do in PBS. And that's just a guess. So what about solving the problem? Well, let's just put in polysorbate, problem solved, right? And if you put in polysorbate 80 at a reasonable concentration, that's the case. At a normal kind of concentration and a super high concentration, you can see that you really inhibit particle formation during pumping. But importantly, during agitation, which granted is a pretty bad stress, you can still see particles forming, microparticles or nanoparticles. And the idea is that surfactants are pretty good stabilizers, but they're not perfect, particularly if you're using a high resolution method like particle to determine aggregation levels. These are really tiny masses of protein being converted to microparticles. And, by SEC, you would say that agitation is doing nothing in the presence of polysorbate, but it is making a lot of particles because polysorbate stability is not perfect. So what's the mechanism for stabilization? Well, proteins are surface active. They tend to go to interfaces. And if you agitate proteins without surfactant, these proteins make films, films break apart, and you get particles. As you put surfactant in with your solution and an antibody, you will find that the surfactant competes for this interface and you get many fewer protein molecules absorbing. Again, it's not perfect, but it's really effective at keeping most molecules off the interface, and you can greatly reduce interfacial stress-induced aggregation. Now, one last comment on this, and this is on analytics. Unfortunately, light obscuration is the dominant method used to monitor particles during studies like process development studies, and also particularly for lot release characterization due to pharmacopoeia methods with arbitrary levels of particles that are acceptable. Light obscuration is a lousy method to detect and quantify protein particles. And here's just a couple of examples. This is looking at studies of microparticles as a function of pumping time. This is that same study data I showed earlier. In some cases, you can pick up things okay with high AC, yeah, kind of maybe, with light obscuration. But here's some really scary examples. Here's light obscuration post-pumping agitation. It's kind of telling you, hey, nothing's happening. The reason this is scary is if you have only light obscuration data, you may have something bad happening in your manufacturing, have huge amounts of microparticles. And there's actually publication from the FDA on an example about this, to where scale up upon commercial product manufacturing led to huge amounts of particles. They weren't noticed because only light obscuration was used. That product ended up killing 7 people and almost killed 42 others. That product was pulled for the market. The company went out of business. And the FDA did a follow-up study, and they learned that there were a lot more micro and nanoparticles in the commercial product than the safe clinical lots. They didn't conclude that the particles were the cause. They didn't have data for that directly. What they concluded was better analytical methods need to be used to characterize products, especially upon scale up or manufacturing changes. So really important, measure particles properly. At a minimum, use a proper method for microparticles. And light obscuration, you might have to use it, but it should be data should be double checked with the method that actually works for protein particles. You learn a lot with nanoparticles if you're able to look at that, definitely during process development. And then for our work, we learned that this post-pumping agitation was a really cool way to accelerate damage and to see differences between formulations. So conclusions: protein stability and aggregation are governed by conformational and colloidal stability. The Timoshev mechanism, Wyman leakage function, can explain protein stabilization in solution during freezing. During drying, it's water replacement. And then if you switch over to interfacial stretch, which is ubiquitous in the life history of a protein drug product, film formation and rupture is the main reason that you get particles. And particle measurements are the most sensitive measure of damage due to things like interfacial stress. Surfactants are helpful, but they're not perfect. And as we all know, polysorbates themselves have their own problems due to degradation. And the last conclusion is read the literature. Even the old literature, things published in the 80s are actually pretty good still.